have a spotlight presentation here in this room. It's going to be given by Stefan Hyde. He is an automation manager at Asahi Kasei Bioprocess America, and he's going to talk about an introduction to OCELOT System Control.
Great, thank you. Hi everyone, as he mentioned, my name is Stefan Hyde. I am the automation manager here at AKBA, Asahi Kasei Bioprocess America. I've been with AKB for a little over a decade, so I'm quite passionate about what we're doing, and I'm really excited to give you this demo of our OCELOT System Control platform. Just a little bit about us. At AKB, our slogan is pioneering biologics safety and manufacturing efficiency. Of course, as an OEM equipment provider, we are doing that by enabling you to do your work more efficiently and more safely as pharmaceutical manufacturers, which really helps all of us in the end. At Asahi Kasei Bioprocess, we have three primary divisions: purification, fluid management, and biosafety testing services. I'll speak primarily about fluid management today. That will encompass all of our automated equipment and pretty much anything that you see that's stainless steel on our website. So the fluid management division is dedicated to solving product safety, efficiency, and purity challenges within the industry. We hope to advance the manufacturing of critical drug substances around the world. And we do that by providing technology in a number of different areas, including virus filtration, inline buffer formulation, chromatography, oligonucleotide synthesis, as well as bioprocessing systems, columns, and bespoke automation solutions. Specifically today, I'll talk about oligonucleotides. We firmly believe that these are one of the most promising therapeutic modalities for today and the future. We've been servicing this industry since 1998, and we have an industry-leading portfolio of equipment, and some of it is mentioned here. Now, kind of to the meat and potatoes, to talk about OCELOT System Control. One note is that today I'll talk about a number of features that are relevant to all of our system types, but of course the focus will be on the synthesizer. Just to start off, our base platform is really targeted to meet all of your compliance and integration needs. We are firm proponents of the Pharma 4.0 initiative and also participants in the Bioforum workstream for plug-and-play systems. It is our desire to be your plug-and-play partner in the industry. Of course, we'd like to sell you all of the equipment that you have in your suite, but we understand that that's not the reality, and that means that we need to play nicely together. I think one part that's often left out here is the IT piece, and so all of our systems are Windows-based, generally Windows 10 Enterprise, but also Windows Server Edition on a virtual machine. Windows 10 can be virtualized as well. But the fact that we're Windows-based and we use Windows security groups allows us to be Active Directory ready. That means we can plug in directly to your existing security system, and your user privileges can be just added to our systems. And that also enables network printing and data export once we're on your domain, as well as scheduled backups or data archival. And the last bit is that we use the Windows time for all of the system timestamps in our systems. And so that allows you to do very simple time synchronization with the rest of your plant. On the OT side, we are PLC-based, Allen Bradley Rockwell Compact Logics or Control Logics. We also offer an option that we call OPC Ready, where we provide a dedicated Ethernet port for your integration. And that could be Pi Historian, it could be controller supervision from your DCS, so that could be remote request of recipes, start and stop of batch. And it can also be direct messaging with other PLCs or control platforms, such as Siemens PCS7, Delta V over Vim. So we can, even if you don't have a large plant DCS, we can send direct messages between those different system types. I will talk about some general functions that I think are just kind of base expectations for a process control platform. First of which is an alarm screen. We, of course, display all the currently active alarms and warnings to the user. You can print this screen as a PDF. And we also demonstrate the last few recent alarms on the process control screen. Those alarms will also be included in an audit trail. The audit trail is 21 CFR Part 11 compliant. You can filter this audit trail by message type, date and time, operator or tag name, and you can also print this as a PDF. This is the first of 3 tools that I'm going to talk about that is in the browser. We have a centralized data server on the system, which allows you to access this audit trail screen from any point in your plant, or a VPN if you're working remotely. So your process engineers can be working on reviewing audit trails. You might want to look at the last 6 months of a specific process alarm occurrence. They can also edit recipes remotely, and they can review batch reports. And all of that can happen concurrently with system operation. We do not charge individual licenses. There is no concurrent user limitation, and there is no specific licensing requirement. Of course, we have a trend screen. You can trend live and historical values. You can create custom groups of pens, update your chart properties like axis labels and colors and things like that. You can also add annotations as the batch is running that will be presented in your batch report. And then, of course, we have a process control screen, so you can review current batch status. You see in a manual environment, you can individually manipulate pumps and valves and associated components. That level of control is also available to you during auto batch if you go into a paused state. So if you had an alarm that you needed to resolve by opening a couple of valves and venting some pressure, that is also possible. But primarily, you're going to be looking at the screen to watch your auto batches execute. And in that environment, you can also interact with your controller set points, as well as alarm thresholds and things like that. And of course, we display the operational status of the components, and the batch status is in the header. Okay, I'm going to talk most extensively about the recipe editor because that's where I get the most of my questions. We'll start here at the Method Explorer. It looks like many other kind of modern file browsers that you have. Google Drive might be the most similar. You have an option for each recipe to recycle, move, or duplicate. And you can also filter these recipes by last edited or created dates if that makes it easier to find. You can create folders. And you can also select them and then preview the recipe. In the synthesis environment, we have kind of 2 tiers of parameters. One is at the sequence level. We call those blocks. And then below that are the sub-steps. You can, of course, double-click or click the pen to edit the recipe. Within the recipe, you kind of have 2 tiers of parameter types. The first is method parameters, and those apply to the recipe on the whole. So anything that is relevant to your entire batch lands in method parameters. Method parameters are also available at point of use when you download your recipe, so you can make modifications to that specific instance. Each parameter that you interact with inside the recipe editor gets a little helper window on the right, so you get your details of your range in units, some documentation about the parameters, etc. In this case, we have a bunch of inlets. You can name those inlets, and those names will show up on the process control screen, and also in your inlet selection drop-downs within the step parameters, which we'll talk about next. Within these blocks, you have a subset of individual steps. You'll see that preview button comes up in a number of these screens. So you can see a brief overview of what exists within this Amidite Edition block. So within these 17 steps, we have a variety of different operation types for your synthesis. Each of these steps can be edited, deleted, or duplicated from this menu, and you can also click and drag. You'll notice a slight delay there, which is really for touchscreen friendliness. So a lot of the buttons are a little bit larger, and there's a slight delay in the interaction when you're dragging things. A couple key parameters. So in a typical synthesis phase, you have, of course, step regulation, which can be by time, volume, or column volume. You have your controller set points, which are enabled dynamically by the recipe editor, depending on which inlets are available or in use by the operator. Your flow rate set points can be by volumetric flow, linear velocity, or also by percent of total ratio, percent of total flow. And additionally, you can regulate your steps by any process value that is relevant on your system. So, maybe most typically UV. So you can either end your step, prompt your operator, or pause the step based on this feedback. So in this example, maybe if your UV is greater than 1.2 absorbance units, after 15 seconds into your recipe and a stabilization delay of 10 seconds. So this allows you to kind of filter out some noise as well that you might have at the start of the step. Also within step parameters, any of the values that you see in your recipe, you can parameterize as a control parameter, and this will allow you to then link those parameters across different blocks. So in all likelihood, your deturbulation flow rate is going to be the same for all of your different Amidite editions. And this allows you to basically create a variable that will assign a value to each of those flow rates. So in this case, I create a variable called dflow p2, and that and you can see that that numeric value gets replaced with that variable. These are useful in a couple of different ways. One is, of course, for just expediting your editing process, but secondarily, these parameters become available to you at point of use when you go to download your batch. So that is nice, one, because you can make modifications if you need to, and two, because it provides an opportunity for some tiered security. So your process engineers are editing recipes here, but you might have an operator that needs to modify some detailed parameters at the point of use, and this will allow them to do that. And it allows the engineer to control what those parameters are. Once that control parameter is created, you can navigate to another step. If you select a relevant variable, it will be available to you, and you can allocate that to that value. Take a second for the video to get there, but we also give you an editing menu where you can change all the default values and the descriptions of your existing control parameters. This will be redundantly displayed in your batch initiation screen. And then finally, you get a library of everywhere that parameter is used, so you know what that is going to apply to. So you see a kind of drill down effect for each of the references. In the step and block editing environment, you can also create favorites. So you can create a favorite block or a favorite step. So any one of these 18 steps could be a favorite, but in this case, the entire block on the whole can be saved as a favorite. You can then use that and build a library of favorites that will be available to you as you build new recipes. So this, of course, can be used within the same step as well. When you add a block from a favorite, it becomes a new instance. So think of it like save as. The difference here is that if I were to just duplicate the T block, any edits that I make to the first T block will apply to the second T block. So all of those are congruous. These kind of save as blocks will then require a distinct icon so that there's no operator confusion. It will also require a distinct hotkey. So the intent there is that you can just type, ACGT and your recipe gets built, or in this case, Tides. We, of course, don't want to have two hotkeys assigned to the same block. Similarly, you can use this recipe function, and this will give you a library of everything that currently exists in the recipe for you to duplicate as a new instance. So those two blocks that have already been created are available to you for copying. And of course, even the first time that you're building your synthesis, once you've built one of these blocks, it's pretty simple to build the next one. All you're going to change is your coupling inlet. So you can go through and add blocks this way as well. Once you have your kind of library of all the blocks that you'd like to use, you can just type using hotkeys. You can also select and duplicate them, and you can delete individual instances. This works the same way as any of the other screens, where you can also just drag and drop, and you can preview the details within the blocks. There will be a brief demonstration about what I was talking about. So we have 4 T blocks here. I say I added this one at T, which is position 7, and I added the titration column volume limit. Then we can navigate to another block and see that update is passed through. That will not apply to the I, D, E, and S blocks unless we've used a control parameter to explicitly link them. Okay, in addition, you'll notice that all these blocks are numbered and they have this little helix icon. That's because we've designated them as monomer additions. But you can also do things like setup functions or shutdown functions where you might be purging, priming, blowdown, things like that. And so you can create blocks where we don't check that monomer addition, and that means we don't add a numeric value, and we give it a little icon that is a valve instead of a helix. Just to give your operator some differentiation. So if you plan to build a 15 mer, you want to see 15 numbers, right? And then in our chain building process, you'll see only the numbered blocks, or only the monomer additions get numbers. Here's a little bit of detail about individual step editing, so we'll take a look at that. You'll see by default none of the inlets are enabled, but as I select inlets, then the flow controllers become enabled and those parameters are available to me. As I said, you can drag and drop blocks around here, and here you see an example of that non-numbered block, so we can move that setup to the beginning. As it states, it might be a little hard to read that note from afar, but the blocks are in 3', 5' order, which is the process order for the system, but we do have a note that they're numbered in 5', 3' order, just to prevent any confusion. So you see the blocks as they will operate on the system, but of course they're numbered correctly for your synthesis chain. Once you have a recipe that you like, you can go to the batch initiation screen. This looks very much like your method explorer. Again, you can have a preview, you can have folders, and you can also sort by your last edited date if you're trying to find your recipe. You get a recipe preview, so you know that you've chosen the one that you want. If the recipe has saved a draft, it will not use that draft copy for iteration until you have committed a new copy by clicking the Save button. You can edit your method parameters at point of use. This is maybe a little bit more relevant for purification. This particular example where your bed might be a little bit longer today than it was yesterday, your pack looks different, and you can edit it now, and then all those column volumes will dynamically update for your recipe. And then again, control parameters can be updated at point of use. So anything that you've designated as something that you want to be able to edit here, you will be able to edit here. The first piece of information that we'll take from you and for your batch is the batch name, and that's how we'll store it in the SQL database and also as a PDF. And then we have a configurable screen below where you can add any information that you'd like stamped on your batch report that you'd like your operators to enter. And we can either make those fields mandatory or not, depending on how you'd like to operate. This last screen is not particularly interesting for an island of automation, but for those of you who are interested in DCS control, these are the tags that we're downloading to the PLC. So we can also take recipes directly from a DCS if you were gonna save all of those recipe values. Generally, we would recommend that you just send us a numeric index or a recipe name, and we can download that recipe automatically in a scripted fashion. And that way you don't have to send all these parameters. You can just send a 4-digit integer, and then the system will do it for you. Okay, once the recipe downloads, it'll take us to the process control screen. The start button becomes available, and I can start my batch. Of course, it'll require an e-signature, but you'll notice also that my inlet names have been added to the flags, and that my batch information is aligned in the header. You see your components start to highlight as the flow paths are opened. Elapsed step time will not start until the step has been initiated, and so you get an accurate delivery time for your amidites. Additionally, we provide a batch status screen. This looks a lot like your recipe editor, so we're trying to give you some continuity here as you're operating. So you can see right now it's running, we're in the T block, we're in the detritylation wash toluene phase. And I can choose to jump to any other phase within that block. I can also jump to another block, sorry, the previous block or the subsequent block with these buttons here. And then finally, I think there's just a brief demo of the trend screen. And we plot 1 point per second as a default. And these values are, of course, simulated and not particularly interesting. The final piece, of course, is the batch report. Once your batch is completed, you want to be able to generate a report. You can, similarly to the trend screen, you can generate chart presets, individual pen properties. And then here is a kind of what you see is what you get chart or batch report preset editor. So I've selected the batch. I can add or remove my recipe information from that report. I can add or remove the audit trail, the charts, choose my chart preset, print it as a PDF, export to CSV, and I can save this template as a default. All the batch information that we entered in that last screen is there, and then you can have also snapshots of volumes. So for example, you could have all of your individual inlet totals for on a stepwise basis or on a method basis. So you get a recipe preview, looks similar to your recipe editing screen. You get a preview of your charts, which of course have simulated data. But you can also kind of observe your chart data here. We will give you a little, some interactive tools on this menu that you won't have once it reaches the hard copy report. And then finally, in the audit trail, you have the same options as you do in the audit trail screen, where you can filter by message type, tag name, or description. Also, you can normalize times to the start of the batch. So we will display all the events from the start to the finish of the batch. If you normalize times, the first time in your audit trail is going to be time 0. And if you don't, then you get the full timestamp, so full date and time, in whatever format you have selected for your system. And that, in total, is OCELOT System Control. I'd be happy to take questions. I'll also be at booth 445 today and tomorrow. Detrip monitoring, you mean in terms of like a UV trigger?
Or just the signal, does it quantitate and integrate under a peak or anything like that?
Not currently, but that is a current development for us. Are you talking about automatic efficiency calculation?
Yeah, some kind of rough estimate of how the one went.
Yep.
Some kind of like report feature.
Yeah, for each distillation cycle, you get an efficiency calculation. It's in the works and not available now. Any other questions?
Stefan, can you, what I like about it is it has the automation functions that an automation engineer needs, but also gives the simplicity of the sequence that a chemist wants to see. So I kind of like that combination.
Thank you.
You mentioned it's Windows 10 enterprise based. So can you talk a little bit about your experience or challenges with, for example, computer system validation, especially if you're tying into the Active Directory and whatnot at the manufacturing site? Have there been challenges there, or has that been pretty straightforward?
So these days, Active Directory integration is really quite seamless. So the applications that we build on either Factory Talk View, which is a Rockwell product, or iFix, which is a GE Prophecy product, they both have excellent Active Directory integration. So as long as we've configured it to use AD groups, then it's a relatively simple process. So in iFix, that's one extra step of synchronizing users. In FTView, there is no extra step. Once that user exists in Windows, that's the privilege that we use. So that has been a really seamless process for us. Yeah.
One of the ways are they're setting up a batch run. So you pointed like you can click and drag or type in like a key, but is there a way to like say import from any some kind of other file?
I don't have a way to import from another file. I wonder what type of file you might be interested in.
Like an Excel file or Notepad file?
Oh,
To avoid any like sequence transcribing mistakes.
sure. I can certainly envision that possibility. It would be relatively trivial, but not at the moment. I don't have that function. So this is for example, as you're scaling, you already have a report, or you have a CSV that you'd like to then. You have a text string or something like that we could import and create that chain. Sure. Yeah. Anything else? Thanks for the time, guys. Appreciate it.